Hello everyone. Today we will be talking on antibodies and immunoglobulins. They are the same thing. Antibodies and immunoglobulins are used in place of each other. Now, how the name immunoglobulins came into existence? Since these immunoglobulins or antibodies are proteins produced by plasma cells and lymphocytes, and they play essential role in body's immune system, and they attach to foreign substances and assist in destroying them. So they are the simple proteins, glycoproteins, which are not soluble in water, and they are the ones which protect us. So they have an important role in immune system. That is why they got the name immunoglobulins. And how they got the name antibodies is when these substances are formed in the serum and tissue fluids, and they respond to an antigen. Antigen is the foreign particle that is entering, and when they a foreign particle entering, enemy entering, or your own cell sometimes start behaving as an antigen. There are different periods. So we will read antigen also in detail. When these immunoglobulins, they fight against antigens. So antibodies and antigen are opposite of each other, and they react with antigen. These immunoglobulins or antibodies react with antigens very specifically. For each antigen, there is a specific antibody. And these reactions are usually in observable manner that we can calculate, we can see, and we can observe either through color, change in color, or there are many ways in, and there are many types of antigen antibody reactions that we will be reading in future. Presently, just understand that these reactions are observed and are readable. Antigen antibody reactions are specific, observable, right? Now, when we are discussing, today we are focusing on only on antibodies or immunoglobulins. So what we have to remember, what is their chemical structure? Chemical structure is, as I have said, that they are glycoproteins. So they have one amino acid and one carboxylic end. So this is the amino end and this is the carboxylic end. Now, these having these two ends, there are, you can see, two chains. One is a longer one, other is a smaller one. So a smaller one, we called it as lighter chain, and the longer one, we called as heavier chain. So every immunoglobulin, they may have structurally, chemically, they may differ, but what they always have is they have two types of chain. One is the heavy chain, another is the light chain. Now, in heavy chain and light chain also, there are certain differences that we have to remember. What are they? In heavy chain, there is this V is standing for variant region. This is a variant region. Variant region, why this is variant region? Because this is arranged like cards. The chemicals which are present in this area, or the amino acids, basically they are the amino acids that are present in this area, can shuffle. They can shuffle and can take the different domains, different shapes. These are variants. Since these can take different shape, that is why there are types of structural differences between immunoglobulins make them into 5 different groups. They are in 5. Only 5 different type of immunoglobulins, or the soldiers which we have appointed, have the chemical structure or receptor to bind with all kind of antigens. Antigens can be hundreds of types. They can, every time, even mutations are taking place in microorganisms, so antigens are always changing. But these same antibodies are fighting against all type of antigens because they have this variable region. This is the variable region in the light chain as well as in heavy chain. So you can see VH, VL in this, and VH and VL here. These are all variable regions where antigen binding sites are present. These are the ones which shuffle and reshuffle and create a domain where antibody fights with an antigen, or the binding of antigen takes place. So remember, these variant regions are very important, and even a heavy chain have a variant region and a light chain have a variant region. So this variant region is the binding site, while the other portion of the immunoglobulin, that is the other constant portions, and all these, remember, all these are joined by disulfide bonds. All these are joined together by disulfide bonds. So these extra things which are constant are not extra. They have biological functions. They play important role in complement activation. What is complement activation? How it takes place? You can see all this in my description box, because I have delivered lectures on that. Right? So those who are missing this can always go in the description box and check the links that I have put on complement activation. While at this stage of lecture, remember that these constant regions are the regions which are actively involved in other biological activity, while antigen binding sites are only variant regions which can shuffle and reshuffle. So they are the portions, they are the domains where antigen can bind. And if you want to, whatever I have spoken is all written here, like when I have said everything in the diagram, and if it's not clear, you can read this slide. So it's all there in this slide. Now, let's do another thing. Let's digest these antibodies with different enzymes in the in vitro system. When we are digesting this with different enzymes, first is papain. When we are digesting this with papain, what will happen? This antigen binding sites will separate out. So we will get two. From here, these antigen binding FAB portions, both the portions of variant region will separate out, and we will give another third portion will also separate out, that will be called FC region. This FC region is the crystallized part which acts on complement activation, which brings about complement activation and other biological activities that we will read in future, right? While when we digest this immunoglobulin with pepsin, what we will find that FC region is completely digested, no FC region is remaining, while antigen binding sites will be there, but their sulfide bonds will not be broken up. So we will have two antigen binding sites, but they will be joined. That means variant region of both the light chains, because every immunoglobulin has two light chains and two heavy chains. So these two heavy chains and two light chains variable regions will remain intact while we are digesting it with pepsin. So these are important MCQs questions. Who are going for entrance exam should remember this. When pepsin digestion is taking place, what is happening? And we are digesting these enzymes because these enzymes are also present inside our body. So when they may be showing these kind of activity inside the body also. So we remember what portions of antibodies are surviving in presence of particular enzymes. And when we do a marketoethanol reduction, we find here, you can see, we find that alkyl portions are all available while the other things have digested. So these are the few things which happens in the presence of certain enzymes. All that I have discussed in the previous slide and I have explained. If you have missed out something, please go through this slide, you will understand. So it is the same thing which I have explained, and then I have put in notes also. I'm putting all the notes, everything there, so that if I'm not able to make you understand properly, if things are not going to you directly by my making understanding it, you can go through my notes and you can relate the two things. So you can read the notes at the end of the lecture too. While sequencing of heavy chains have also done, and these sequencing has revealed that 100 to 110 amino terminals that I have already told you, NH3 terminus, which I have explained in the slide where I showed the structure of immunoglobulin, is highly variable. That means 100 to 110 amino acids are highly variable. And this shows 5 basic sequence patterns. When they were worked out, 5 basic sequences have been found out, and they have been named as alpha, gamma, delta, epsilon, and mu. So as they have been named on the Greek letters, they were in English letters, they were like Ig was standing for immunoglobulin, I capital, G small, and then alpha A, IgG is gamma, IgD is delta, IgE is epsilon, and IgM is mu. So the above classes are also called isotypes of them. Each class will have either a kappa or delta light chains. When we were talking about light chains, let me remind you, light chain can either have a kappa structure or gamma. So either a light chain or in light chain, either a kappa or a delta will only be present, right? All the light chains will not have kappa and delta together. So either kappa will be present or delta will be present. That should be clear to you. Minor differences in IgA and IgG have classified it into IgA1, IgA2, while IgG is classified into IgE1, 2, 3, 4. So these are the ones which have been classified. Now, different classes of immunoglobulins are there, and on the basis of these classes, they have been classified, and the way they react with antigen, they interact with antigen, they have been classified as IgG, that is gamma, IgA alpha, IgM mu, IgE epsilon, and IgD delta. For you to remember, just remember that they are gamete. Here I am going to end this class with the types of different immunoglobulins, that is gamma, alpha, mu, epsilon, delta. Each I will be explaining in the next class. So these two will be related together, but I don't want it to be too static and not interesting, because it will become too long. So I just hope that you go through this, and in continuation you take my next class also. If you like my channel, please do like and then subscribe it. Thank you so much.